That's good?
Yeah.
All right. And first, I would like to thank Professor Yao and Dan for inviting me. And so what I would like to share with you today is some of the challenges and the opportunities and we have encountered in the last few years and when we work on the massive genetic genomics data. So this is more presenting the questions than presenting the full solution. Okay, so I'll start from the introduction, and people probably heard about omics, so I'm going to change the word to om, so then I'll explain why. And so why we are living in this exciting time of big omics data and what opportunities and challenges. And then I'll talk to illustrate the point, and I'm going to focus on a few areas. One is a whole genome sequencing association study. The second is multiple phenotype analysis, and third one is analysis of integrated different type of genetic genomic data from different platforms, and then finally I'll present the discussion. And so many of you probably heard about the Human Genome Project, which was launched about over 10 years ago, and the Broad Institute was the leader in this initiative. And so the massive after the Human Genome Project, and this really has translated the landscape of biomedical research. And in the last few years, massive genetic genomic data have been generated. And in recent years, and the genome data have been extended more to collect the exposome data. And so this is massive exposure data. And also phenome data, which is massive phenotype data, in statistician called the outcome data. And so this massive OM data have really transformed the basic clinical the population science research in helping us understanding the disease biology and also the interplay of gene and the environment in causing the human diseases, and also the advancing the precision medicine. You probably heard about the personalized medicine, but this is a more advanced concept than personalized medicine. So basically trying to identify subgroup of people and where the prevention strategy and also the treatment strategy can be effective. So we call the precision prevention and precision medicine. And so in the last decade, and different type of genetic genomics data have been generated. In early days, and was like a gene expression data. And in the last few years, in the last 10 years, and so people probably heard about the genome-wide association study. And so those are the genotype data. And then in the recent years, and more epigenetics data, such as the DNA methylation data, have been generated across the genome. And so the most state-of-art technology is a whole genome sequencing, which includes both the DNA sequencing and also RNA sequencing. And so if you compare the what has happened in the last 10 years is really dramatic change. So for example, if you look at the blue line, that is the Moore's law, which captures the cost of computing. This is on the log scale. And so the cost of computing has decreased exponentially in the last 10 years. But if you look at the cost of sequencing, you can see that the cost of sequencing has dropped dramatically, even faster than the exponential rate. And so about over 10 years ago, and the cost of sequencing the whole genome is about $3 billion. And nowadays, so the goal is to have about $1,000 genome. We are not quite there yet. Right now, it's about $1,500 to sequence the whole genome. And so the idea is, in a few years down the road, we can reach $1,000 genome goal. And so basically, for a subject, and it sequences the whole genome and costs $1,000, and then this whole genome you can put in a pocket, go to the doctor's office, and one doctor can review your genome information and identify what potential diseases you are at risk, and also what potential treatment which can be effective. And
How long does it take?
Sorry?
How long does it take
to sequence the whole genome? Yeah. Oh, it's very quick. Now it's very quick. And so right now, I think the sequencing itself is not the challenge. The challenge is more in the analysis, in the library building, and also the genotyping call, and also the assembly. And so because of the dramatic drop of the sequencing cost, and so the number of human genome which has been sequenced has increased dramatically over the year, which is captured by this black line. So for example, the National Institute of Blood and Heart and Lung, and so they just launched a new initiative. They are going to sequence over 20,000 people, and for the whole genome sequencing. And so we are part of that. So we are going to have, they are going to sequence about over 1,000 people, and for the sleep apnea. So that is a disease trait we are involved in. And so the Harvard alone probably are going to sequence for the Brigham, and so they sequence totally about 5,000 people across multiple phenotypes. And so the challenge, so this really presents lots of opportunities, and in the meantime, it presents lots of challenges. And so the difficulty and the challenges in analyzing human genome is nicely captured by this Eric Lander's classical code. And so for those of you, the student, probably you heard about this Nobel Prize, which generally is presented every September in the Sanders Theater. And part of that is called this 24/7 lecture. So basically, and the idea is have a subject, and then the scientists need to describe the subjects and in 24 seconds, and using technical terms, so only your classmates can understand. So for statistics, for example, you think like a Bayesian inference, estimation, asymptotics, and so on. And so the most challenging part is not that. Every statistician can do that. The most challenging part is the 7. So what is the 7? 7 means that you need to describe this subject, and in 7 words, your mother and grandmother can understand. And so Eric Lander was invited to the Ingo Nobel Prize lecture in 2003. And his 7 words describing the genome is the genome, about the book, hard to read. And so that describes the genome very well. So one can sequence the genome, but then how are you going to interpret the findings? How can you find out the gene and to which cause the lung cancer? And also how the biology really works and causing the human diseases. And so suppose you are invited to giving this 24 lecture, a Nobel Prize 24 lecture about statistics. And so what are you going to say? You can think about it, and then I'll tell you what one of our colleagues said. And so the 24 is always easy. And what is the 7 words you're going to use to describe statistics your mother can understand? Okay, so this is about genetics. And so in the last few years, so there are more and more data available for exposome and also phenome. And what is exposome? Exposome is a massive exposure data, and which include the social network and also like exposure data, for example, air pollution data from satellite. And those are massive data. And also the historical exposures information. And so then also, what is a phenome? Phenome is a massive phenotype data statistician calls outcome data. And so here give you examples about what are the possible phenome data. One is electronic medical record. So basically, one can use, what the idea is, rather than you are running the large clinical trial, one can use the large administrative database, for example, partner database, and which include all the patient information in the Harvard affiliated hospitals, and then to identify the effective treatment strategy, and which can be effective for a particular disease. And so the data are massive, and also the imaging data. How, for example, how can one connect the genetic genomic data with the imaging data? And the imaging information generally is 3-dimensional. And another one is called this digital phenotyping. And so they basically, the idea is one can measure the digital exposure and phenotype information using the smartphones. For example, for those of you probably heard about this Apple Watch, and so that is in Apple Watch, and there is one called the ResearchNet. And so the idea is one can measure for example, in this my heart count, so one can measure basically continuous heart count for everybody, and continuous over time. And so therefore you can see using those kind of like digital technology, one can collect massive genetic genome, massive phenotype information and exposure information. And also one can encourage more people involved in the study without doing the phone interviews. And so those are really great opportunities, but in the meantime, they really present lots of challenges. And so we faced with this massive data and with this big beast. And then how can we be the beauty who can handle the beast? And so this is really nicely captured by this McPherson's Nature Medicine review articles in 2009. He said, bridging analytic gap is essential, or the convenient $1,000 genome will come up with a $20,000 analysis price tag. So this is great for statistician and mathematician and computer scientists. And so in the next 10 years, there will be plenty of job opportunities. And so just tell you a little story. And Chelsea Clinton was interviewed in our school about last week and in this leadership voice. And so one of the colleagues in school public health and asked her during the interview, the question is, I think one student asked that question. So what is the most beneficial subject you learned at Columbia when you was a student there and in school public health? And she replied in one word, statistics. And so this really a great signals and tell the student in the room this is really a great field. For those of you who have heard about this data science course and as undergrad course here, which was offered twice in the last 2 years, and the year before last year, and 375 students enrolled, and last year also over 300 students enrolled. So there's really a lot of interest and in Harvard College alone and in the data science, and that. Okay, so how can we deal with those massive data? And so generally that involve more than just statistics. So one need to have a close integration between quantitative method and science. And also one needs to develop computational and also statistical methodology. And the easiest part is high performance computing. That so probably students probably heard about this FAS cluster, which is located in Mount Holyoke. And this high performance cluster is shared by 6 universities in Massachusetts and have thousands of nodes, and which allow high performance computing. So this hardware is the simplest one. The challenging part is the first 2 bullets. And so what is the role statistician can play? And for those of you who pay attention to this article, which was published in Science last year, and so this one compares, so the authors are all our Harvard colleagues. And so they compare the Google estimate of the flu incidence with the CDC estimate of the flu incidence. You can see that by looking at this peak here, and Google estimate double the CDC estimate. And so after that, and the massive medium coverage articles have appeared. And so this is nicely captured by this title. So big data, are we making big mistakes? And so generally, it is possible we have massive information and we are more likely to make mistakes. So this also illustrates the role of statistician. How can statistician help? And by reducing the chance of mistakes. So the data science will involve computer science, mathematics, and statistics, and other quantitative science and information science, for example. And so what is the unique role statistician can play? So I would say the inference, so it helped control for errors. And now we can come back to this 24/7 lecture and on statistics. So this one, so the question is, if you are invited to give 24/7 lecture on statistics, if you want to describe statistics in 7 words, your mother can understand, what are you going to say? Okay. And so as a matter of fact, one of our colleagues, Shelley Meng, and who's currently the dean of FAS, the graduate school, and so he was invited to this 2013, and you go 24/7 lecture on statistics. And his 7 words is the only critical ball approved by the God. And so that basically means like statistician can help taking account for the random errors. All right, now go back to genetics, genomics. So what are the major quantitative interests and in genetic genomics? The first one is signal detection. So basically the question is, how can I find the genes and which cause lung cancer? And however, the challenge is the true biological signals generally are often weak and sparse. And so how can we identify the weak and sparse signals? The second area is the analysis of integrated data, which include a different type of genetic genomics data, genome data, exposome data, and also massive phenotype data. And the next one is a network analysis. How can we understand the biological network which causes the human diseases, how the gene work together in a concert. And also, how can we use the data, like exposome data and the genetic genomics data to develop risk prediction and also precision prevention strategies and also treatment strategy. And finally, the reproducible research. And that includes how to make the data reproducible and also how to make the result reproducible. So this is one of the top priority at NIH right now. And Francis Collin had a paper in Nature last year. And so NIH has this major initiative in promoting the reproducible research. And so all the NIH trainees need to have training in reproducible research. Okay, now let me move on to what we are going to focus on. So we are going to focus on signal detection. That is, I'm going to focus on the whole genome sequencing association study, especially for rare variants, and also multiple phenotype analysis, and including the analyze the genetics networks and the field trophy, and also imaging genomics when we deal with the massive outcome data. And also, the second part is how to analyze integrated genetic genomics data. Okay, so signal detection. So what the basic idea? The basic idea is basically we need to look for needles in a haystack. And so this is very nicely illustrated in this cartoon. This gentleman, So this gentleman holding the box of 3 billion pieces. So that is 3 billion base pair across the genome. So the genome total has 3 billion base pairs. And so this gentleman said, I think I found a corner piece. So that is a gene that causes lung cancer. So the question is, how can you find out that corner piece? And so this basically is a strategy. These include statistical strategy, computational strategy, and biological strategy, so that they have to integrate together. Okay, so what could be a good strategy? So in computer science, a good concept to handle massive job is to use this divide and conquer strategy. So basically, if you want to run the massive job, you do parallel computing, and then divide to the different node. So the analysis of massive data, the big data, and can use this kind of strategy. But the question is, how do we divide? How do we conquer? So that is a challenge. It depends on the art, depends on the scientific discipline. And so I'm going to illustrate this strategy using 2 examples. The first example is the simple example, the GWAS example. And so what is a genome-wide association study? So this idea is in the old days, one genotype, one SNP at a time. This is called single nuclear polymorphism. And in the last 10 years, then this SNP array allow us to genotype 500,000 to 5 million genetic variants across the genome. But for the SNP array, it only covers common variants. So what do we mean by common variants? So generally at each loci, that basically is a location on the genome. And so generally the genotype takes the following value, take AA, AB, or BB. So one from mom, one from dad. A is called the wild type, B is called minor allele. And so the coding generally has 0, 1, 2. So basically the number indicates the number of the B allele, the minor allele. And so if the probability of B, that is this is called the minor allele frequency, if it's greater than 5%, so that means the B allele is common enough in the population, this is called common variance. So generally, the GWAS chip covers the common variance. So in this simple situation, the divide and conquer strategy is you analyze one SNP at a time, and then regress the outcome, for example, case control status on each individual SNP, and then you got the p-values, and then you calculate this called Manhattan plot. So plot out the log p-value of each SNP and regression of the case control status, for example, lung cancer versus control, against the genome location. And different color represent different chromosomes. And generally, because one does a lot of hypothesis testing, so a lot of p-values, which are very small, are false positives. So therefore, the idea is you want threshold low. And so, for example, pick up the top 1,000 SNPs, which have a small p-value, and then validate them using the separate validation study data or the meta-analysis. And if 5 years ago, if you can validate some of those top SNPs and using this strategy, you'll be able to have a paper in Nature Genetics. Okay, so now this strategy is much more challenging, and because this field is much more mature. Okay, and so this strategy, this divide and conquer strategy, worked very well in the GWAS setting because the variants are common. And so in the last 10 years, and thousands of genetic variants have been identified across all the chromosomes, and which are associated with different disease phenotype and the disease trait. And so this is illustrated by this NHGRI and GWAS catalog. And also, in the last 10 years, NIH has really pushed for the data sharing. And so, for example, so the open access, like HapMap data or 1,000 genome data, and they are all open access. And one can also access many, many of the GWAS data by filing an application to dbGaP. And because of data confidentiality issues. And so therefore, so if one want to have access to the GWAS data for your dissertation, and so basically you just need to apply for the dbGaP application. You don't need to run the GWAS study by yourself. Okay, so with all those successes, why do we bother with sequencing? And the problem is the GWAS only focus on the common variants. So that is the variants with a minor allele frequency greater than 5%. So that loci is we have sufficient people with that particular B allele and that location. And also the GWAS study has found out the effect of those common variants are pretty small, with the relative risk is between 1.2 to 1.5. And another thing is there's a large portion of missing heritability. What does that mean? So if one uses the GWAS hit to explain the variation of height, and so we know from the family study, and if mom dad are tall, generally the children will be tall. So from family study, we know the heritability is about 80%. So 80% are genetics for height. But if one uses a GWAS hit, it only explains 10% of the heritability. And so that means a large portion of heritability is missing. And secondly, as I mentioned, the GWAS chip only covers the common variants, and it doesn't capture the rare variants. Those are those loci with minor allele frequency less than 1 to 5%. So that's why the state-of-art technology is the whole genome sequencing. So what is the whole genome sequencing? So the idea is one genotype, all base pairs in a gene, a candidate gene, or the whole exome, that is the coding regions, or the whole genome. The whole genome totally have 3 billion base pairs. And so if you think about, suppose you collect data about like 10,000 people, so the data has 10,000 rows, but the column, the number of columns of the data matrix is 3 billion. Okay, so you can see the data are humongous. And so what does GWAS cover? What does the sequencing cover? So the GWAS cover, suppose this probability of T is say 10% in the population, then this particular loci will be on GWAS chip. If this probability of C, say, is like 20%, this is also common variance, and this particular loci will be on the GWAS chip as well. And but the sequencing, so you can see GWAS miss a lot of loci. Sequencing basically gets genetic information, the genotype information of every single location across the genome. So basically, you got information across every single base pair across the genome of the 3 billion base pairs. And so you can see the data are humongous. Makes things worse. Across the genome, a vast majority, over 90% of variants across the genome are rare. So this is a histogram and of the minor allele frequency. So if I zoom in and to minor allele frequency between 0 to 5%, you can see that lots of variants are very, very rare. So this is based on NHLBI exome sequencing project, and they sequence about over 8,000 people for the coding regions. And as a matter of fact, many of those variants are private variants. So that means only you have it, I don't have, nobody else have. So that makes the analysis much, much more challenging. To illustrate, so I'll illustrate using this early candidate gene study. And so in this study, in Dallas Heart study data, and they sequenced 3 candidate genes, and totally they sequenced 3,500 people. And you can see the sample size looks big, right? And so totally among those 3 genes, there are 93 variants. And so what is interesting is among those 93 variants, 50% are singletons. That means only one person has it. Nobody else has it. And so the goal of this study is to identify the effect of those genes on the lipid trait to show you how sparse the data is. This is a heat map of the first 500 people. And so totally 500 rows indicate 500 people. And so totally there are over 3,000 people here for illustration. I only illustrate the first 500 people. And then totally there are 93 columns because this is candidate gene study. So in the whole genome sequencing study, there will be 3 billion columns. Okay, so if you look at the heat map, the black indicate zeros, okay? Black indicate 0. The black spot indicates zeros. Bright spot indicate 1 or 2s, okay? And so you can see only in this particular loci here, and so there's a lots of bright spot. So that indicate this particular loci is a common variance, okay? But if you look at all the other locations, and you can see a lots of black spots. As a matter of fact, for many of those columns, 50% of those columns, you only have 1, 1. All the rest are zeros. So you can see this design matrix is very, very sparse, okay? And so that create a lot of challenges. And so because the design matrix is so sparse, for each column, you have a lots of zeros, only very, very small number of ones. And so the analysis used in the GWAS strategy, so basically the divide, that means you analyze one SNP at a time, conquer, then you run regression by analyzing a run SNP at a time. Doesn't work anymore. And and furthermore, if one analyzes one SNP at a time, and there's very little power because there's very small number of people who have the variants. And so therefore, the strategy, the divide-conquer strategy had to be changed. So one needs to sequence the genome beyond the single SNP analysis. So one possibility is to scan the genome and using a SNP set. So for example, you divide the genome based on the gene, and but when you use exome sequencing, this works because the coding region is within the gene. But if one sequences the whole genome, because there are many intergenetic regions, and so this gene level analysis doesn't work. So one can think about the sliding window strategy. So that basically means one analyze moving the window, like for example, 5,000 base pair at a time, then moving across the genome. And then one can also use the pathway, genetics pathway as analysis unit, or genetics network as analysis unit. But if one uses this moving window and to scan the whole genome, and there is a challenge, what is a window size? Should you make the window size if 5,000 base pairs or 10,000 base pairs? So if you make the window too small, there are not enough variants. If you make the window too big, there are too many chunks. And so this is a challenge. And then also after, suppose you identify the region you want to analyze, and you do the moving window strategy, and then you can test the whole SNP set effect that tests this one gene, the effect of the gene. But each gene, they can be hundreds of variants or thousands of variants. And so this can be put into the statistical framework. Suppose I have a covariates I want to control, like age, gender, population stratification. And also within, suppose I analyze one gene at a time or one region at a time. Within this region, I can have 100 variants, or I can have 1,000 variants. And because some genes are big, some genes are small. And also, to make things worse, this genotype matrix, and I call the G here, and so P is the total number of variants within the gene or within the region, and so say P is 100. And then the G is the genotype matrix. So basically what you have is this G matrix, because many of the variants are rare variants, so this genotype matrix is very, very sparse. So that means this matrix has lots of zeros, only very small number of 1 or 2s. And now what I want to do is I want to study whether there is association between the disease phenotype. For example, if I have lung cancer case control status, I'm going to run the logistic regression, regress on the covariates, and regress on the 100 genotype within the gene. And if you have network, this will be the genotype within the network. Then I want to test whether there is overall association between the region or the gene and the lung cancer risk. So I test 100 beta equal to 0. If you have a large gene, then the P can be large, can be 1,000. If you have a network, it can be even bigger. Okay, and what are the statistical challenges? First, the genotype matrix and very sparse. The G matrix is very sparse because the sequencing data, they have lots of rare variants. So the matrix has lots of zeros, very small number of ones or twos. And also, some SNP are in LD. We call the LD called linkage disequilibrium. So in statistician called the correlation, but they mean the same thing. And so we need to account for the correlation structure of the SNPs. And so basically the correlation among the columns. And also the number of SNP in each region or the genes or the network and may be large. And so suppose I have like 1,000 SNP within this gene. If it's very difficult to fit this model because this genotype matrix is too sparse. If you have a binary outcome, lung cancer case control status, and because the genotype matrix is so sparse, I can even not fit this model. And the model will fail to converge. Secondly, and because I test so many parameters equal to 0, 100 parameter equal to 0, or 1,000 parameter equal to 0, and if I do the traditional likelihood ratio test or word test, I don't have power. Okay. So the question is, how can we how can we borrow information across multiple SNP within the gene? And to make things worse, another challenge is alternative hypothesis can be dense or sparse. So what that means is, how many of the betas and are really not 0? And so think about this situation. If I have 100 variants, if God tell me that only these 5 variants, 5 SNP are associated with disease, then basically I need to pick up those 5 and do the regression, right? But in reality, I don't know which 5 are associated with lung cancer. I cannot do that strategy. So in this situation, that means 95 of the beta are 0, 5 of them not equal to 0, right? And so this is called the sparse regime. And the dense regime means majority of the beta not equal to 0, okay? So to illustrate, suppose I make the K is the number of causal SNP. So that is the 5 SNP which are associated with disease, that 5 beta not equal to 0. P is the number of SNP within the gene or the network. And then I let the number of true signal is the function of the P, that is 1 minus alpha. And so that is the K is the number beta not equal to 0. So you can see that if alpha equal to 0, then that means that the data are very sparse. If alpha equal to 0, data are very dense. The signal are very dense. If alpha equal to 1, then you can see the signal very sparse. So bigger alpha means the sparse signals. And so give you an example. So my hypothesis, I want to test all the betas within the gene equal to 0. That is 100 beta, 1,000 beta equal to 0. Alternative is some beta not equal to 0. I don't know which one. Okay. So the dense alternative is when the alpha less than half. When I say dense alternative, it doesn't mean 50% of beta not equal to 0. That means this power alpha is less than half. So if I make the P equal to 100, if I make alpha equal to 0.6, then K is 16. That means 16 beta not equal to 0, and 84 equal to 0. And for the sparse alternative, that means alpha greater than half. So if I make the P equal to 100, alpha is a 0.4. This is a sparse regime, and the K equal to 7. That means 7 beta not equal to 0. So the optimal strategy depends on whether the alternative is dense or sparse. Okay, and makes things more challenging is so there's no global optimal test in this setting. And so the optimality of the test will depend on the genotype matrix G, how sparse the genotype matrix is, and what is LD structure, how correlated the SNPs are, and also depends on the signal, beta. So how sparse the signals are, and how strong the signals are, and also whether the betas, non-zero beta, are in the same direction or in different direction. Okay. So because people don't know how to deal with such kind of data, people say, how about we do something simple? I'm going to do dimension reduction, and what I'm going to do is I have 100 variants. I don't want to fit a model with 100 parameter. I'm going to clap the variance. So I'm going to count how many real variants each person has. And then that will be the dimension reduction. So for example, if I use this toy example, so those 1 is a Y, those are the lung cancer cases, 0 is the controls. So those, suppose I have 4 SNPs. So the first person, you can see there's only 1 real variance, and then the dose will be 1. And the second person, there's only 1 real variance, so the dose will be 1. And the third person, there are 2 real variants, the dose will be 2. So I do this simple dimension reduction strategy. I reduced from the p-dimensional to single dimension. Great, smart strategy, right? But what's the problem? The problem is this strategy makes a strong assumption. So what is the assumption? The assumption assumes that all the variants in the gene are causal variants. That means all the betas are not 0. And why is that? The reason is, if you look at here, so if all the betas the same, I can take the beta out, I can clap all the genotypes, right? So this assumption basically means, assumption here is all the betas are not equal to 0, and also all the betas the same. If some betas are 0, some beta not equal to 0, or some betas positive or some beta negative, this strategy will fail. The signal will cancel out. Okay. So this is another strategy our group developed. We call the SCAT strategy. So this is called a sequence kernel machine association test. And so this has been widely used in the sequencing association study community. So this is based on the variance component test strategy. So basically the idea is, so this is derived using the variance component test, but turns out it's a very simple form. And so basically, rather than aggregating the variance, we aggregate the statistics. So that means, suppose you have the score statistics and of each individual SNPs and on the disease outcome. So basically, that measures the correlation of each SNP and the outcome. And this is a score statistic. So I square it and then multiply the weight and sum over all the 100 variants. And so because it's a score statistic, I just need to fit the null model only once. So computationally, that's very fast. So when we handle large beta, and so the scalable is very, very important. We need to make it really fast. If make it fast, and then people will use it. And so it turns out the calculation here is very quick to figure out analytic distribution. But in reality, because we don't know whether this burden strategy is optimal or the SCAT strategy is optimal, because in reality, when we scan the genome, some genes, maybe a lot of variants are causal variants. Some genes, and maybe only a small fraction of variants are causal variants, effect may be in different direction. So therefore, because we don't know what will happen, so therefore a better strategy when we scan the genome is this omnibus strategy. So basically, that can also be calculated using this variance component test, but it also takes a simple form. It's a linear combination of the 2 test statistics. And so in order to find out optimal C, so basically one can maximize power to find optimal C, and then use that as a test statistics and figure out the p-value, taking into account the C is estimated. Okay.
So what is WJ?
What?
What is WJ?
Oh, this is where the biological knowledge kick in. So based on the population, so it can be using the functional information. So for example, if you know the variants are non-synonymous variants, and then you can up with them. Okay, and with this is
Designed by the user, input by the user.
Input by the user, or you can up with the real variants. So for the variants which are more rare, you up with them more. Okay, that is where the biology comes in. And so, for example, population genetics information will come in here. To apply this and to the Dallas Heart Study data, so you can see that different method works better for different genes. So for example, like the first scenario, you can see that the burden test works better than SCAT. And the SCAT, this optimal strategy, try to be closer to burden. And so this C, you estimate from the data. But if you look at this gene, you can see SCAT work much better than burden. And this SCAT, omnibus strategy, try to be closer to SCAT. Okay.
Are those genes we know to be implicated in the phenotype in this case?
Sorry?
Were those genes selected because we already know that they're part of the phenotype?
Because those are candidate gene study. This is an earlier study. Yeah, right. Yeah. Okay. And so this is a dense alternative situation. Now let's look at the sparse alternative. So the strategy I showed you before worked well if we have signals are dense under the alternative. But in reality, you don't know whether signal are dense or sparse. So as mentioned, if I have 100 variants, if I know God tell me only these 5 are associated with disease, those 5 beta not equal to 0, and I should take all those 5 and throw away those 95. Those are junks, right? But we don't know that in reality. What can we do? So the basic idea of this strategy is throw away the noise snips and optimally keep the signal snip. But then the art is how can we keep the optimal signal snip? So this one idea was this hierarchism idea which was proposed by David Donahoe and Jiasun Jin in 2014, and following the original idea of Tukey. So the idea is we want to threshold the test statistics by some constant C, and so then we aggregate them. But then what is the constant C you should use? So if you use the p-value 0.05 to threshold, that is not a good strategy, okay? So then the art is how can you find out the C? But the challenge here is, so that is basically this hierarchism HC strategy. But the key assumption they make in that case is they assume all the tests are independent to each other. This doesn't work for genetic study because for a gene or network, and I know the SNP are correlated to each other. So I need to account for the LD structure. So this is what we propose. We call the generalized hierarchism strategy. So basically that account for the correlation among the SNPs. So another strategy is for the stat students who have followed the variable selection literature, you would think, what will happen if I do the sparse estimator, use those as a strategy? So for example, I can do the variable selection strategy and using like lasso, SCAD, and which is based on this Pinot likelihood strategy, and estimate the beta hat which is not equal to 0. And then use those to construct the test statistics. And so what happened if one use this strategy? And so this, ideally, you think that will work better. This help you throw away the noise, keep the signals. And so if you use this strategy based on the Pinot likelihood, what happened? It turns out the story is challenging. So if you let both n and p goes to infinity, this is where the mathematics helps. And when deal with a high-dimensional data, you let p goes to infinity, n goes to infinity, but they can have a different rate. It turns out that if you use this variable selection strategy, if you have a consistent variable selection strategy, for example, like the SCAD strategy or the lasso strategy, the asymptotic size is 0. One cannot control for temporal error as a 0.05. Furthermore, the detection boundary is the same as the hierarchism detection boundary. And also, if one considers the local alternative, statisticians are often interested in hypothesis testing. And this Pinot likelihood test has no power. And also, another challenging thing is, so this result applied to linear regression when the outcome y is continuous. But when the outcome is binary, like case control status, and then things become even more challenging. And so if the G matrix is too sparse, and like the sequencing data, G matrix is very sparse, and then all the tests can be powerless no matter how big the signals are, okay? So basically there's no detection boundary. And so the detection will depend on how sparse the design matrix is, how sparse the signal is, and also how strong the correlation of the SNPs are. And so you can see this problem is quite challenging for both from theoretical point of view and also from applied point of view. Okay, now let's move on to part 2, the multiple phenotype analysis. So the idea is that one would think a particular gene or SNP could be associated with multiple diseases or multiple traits, like lipid. Generally, they can have multiple traits related to lipid. So one want to identify the SNP which is associated with multiple traits. So the multiple trait can be the imaging, okay? So that can be a very high dimensional. And or can be the gene in the network. And so how can we detect this? We call the pleiotropic SNPs, and using the GWAS summary statistics without knowing the original data. I just know the summary statistics. And because many people don't want to share the original data. So how can we do that? So this related to the dimension reduction. Okay, so a simple strategy is I'm going to do the dimension reduction using the principal component strategy. So you can see for this situation, I only have two variables here. And then suppose if I do the individual Z statistics, and the Z statistic is about 3.6 and 4.4. So this is marginally significant. Now I'm going to do the dimension reduction. So which dimension should I use? If I use the PC1 direction, you can see if I project to the PC1 direction, you can see this won't have any power. So you can see PC1 direction, not a good direction to project, even explains the most variability in the data. If I project to the PC2 direction, and so this direction, and you can see, and then the p-value is much more significant. So the question is, when I have a high-dimensional phenotype, if I do the dimension reduction, do the PC projection, which direction should I use? And so here, I just show you a few possible statistics one can use. The first is you analyze one phenotype at a time. You calculate the p-value, you calculate the minimum p-value, and use that as a test statistics. And or you can use the Fisher method. You calculate individual p-values of the PCs, and then you aggregate them across all the p-values. And or you can use a PC square divided by the eigenvalues. So basically, in this situation, you upweight the small PCs, downweight the larger PCs. And so this works. Those strategies work for the dense signal. If you have a sparse signal, then one need to think about thresholding. So one can think about this generalized hierarchism strategy by optimally combine threshold individual trait p-value. And to show you how this works, without showing the math, because we don't have time, I'm showing you this analysis of this GWAS data for the 4 traits. So this including the HDL, LDL, and cholesterol, and triglyceride. Totally, we have 2.6 million SNPs. And so this basically showed you the number of new significant SNP. So what we mean by new significant SNP? Because I can analyze each trait at a time, right? So I want to see whether I can identify the new SNP which cannot be identified using this individual trait analysis. So this is what we mean by the new significant SNP. So you can see if I do the minimum P strategy, I don't identify any new one. If I use a PC1, not much, and I only identify 19 new one. If I use PC4, I identify 180. But if I use by combining information across the PCs, for example, if I use this PCSS, by upweighting the small PCs, I identify much more. So why that works? In order to see why that works, so basically, one really needs to understand the theory. So the power of the PC-based test depends on the dimension, also depends on the signal sparsity, depends on the effect direction, and also depends on the correlation structure. And also, this is also where the mathematics can help. It's important to understand the geometry. So in order to see which PC works and have the best power, basically one need to calculate the principal angle. So that is the angle of the PC and the true signal angles. If the PC angle and the true signal angle, they align, that PC direction is the best direction. If that PC angle and the true signal angle, they are orthogonal, that PC has no power at all. Okay? And also, this also tells us higher order PC can contribute substantial information about the association. And so if one uses the PC1 that explains the most variability in the data, that may not be the best strategy. Even explain the most variability in the data, but if you are interested in testing for association, that may not be the best strategy. Also, one want to combine optimally the information across multiple PC. So that is the art is how do you identify this optimal combination. So that is where statistical math can help. So the last part is the data integration. This is one of the major fields many people are interested in, integrating different types of genetics data, exposome data, and phenotype data. So here I'm going to focus on the genetics data. By integrating different types of genetics data, one can understand, for example, like the GWAS data or sequencing data with the genomics data, such as expression data, microRNA, or DNA methylation data, or the exposome data, that's massive exposure data, and for example, social network data, and the phenotype data, phenome data. So there are multiple strategies one can use. One is the mediation strategy, and to understand a biological pathway. The other is network strategy. I'm going to focus on the mediation strategy. So what is the mediation strategy? I'm going to, without showing the math, and I'm going to show you the example. So the idea is, so this couple years ago, there is multiple GWAS study which had been conducted to study which SNP associated with lung cancer. They found out 2 SNP on chromosome 15 associated with lung cancer. What is interesting is those 2 SNP on chromosome 15, they are also associated with smoking. So therefore, a natural question is, GWAS study is so expensive, millions of dollars one need to conduct the GWAS study. And whether we find anything new, whether we really just find a smoking effect, right? So the natural question we want to ask, the community ask is, are those 2 SNP and identified using the GWAS study affect the lung cancer through smoking? So this can be answered through this causal mediation diagram. So here is the 2 steps on chromosome 15. This is the smoking, and this is lung cancer. We want to see what is the effect of those 2 steps on lung cancer through other biological pathway, not through smoking. This is called the direct effect, or how much of this effect of those 2 SNP on lung cancer through smoking. This is called the direct effect. And so those we want to control for covariates as well. So by doing this analysis, and then one can show that, so this is based on 4 cohorts. Totally, we have over 10,000 people. And so here is the direct effect. That is the effect of the SNP on the lung cancer through other biological pathway, not through smoking. So you can see by meta-analysis, it is highly significant. But if you look for the effect of those SNP on lung cancer through smoking, you can see it's not significant. As a matter of fact, if one calculates the percentage of effect of the SNP on lung cancer mediated through smoking, it's only explained about 3%. So that is great. So that tells us GWAS tell us a little bit more than smoking. Okay? So this can also be used to integrate different type of genetic genomics data. For example, like the GWAS data or sequencing data with the gene expression data or DNA methylation data, and to understand the biology to see how the gene affects asthma. And to show you, which one? This one?
SNPs are associated with smoking as well. So in other words, for those people who see smoking, really smoking is not causing any kind of...
No, the smoking is a cohort study. This is based on everybody. Yeah. So I'll explain, use this example. I'm going to show you how we estimate mediation effect. I'll explain the smoking result, okay, using the model. So this is how the statistics can help. Okay, so in order to do the mediation analysis, so basically what you do is you do joint modeling. You model you do the probability of lung cancer risk, do a logistic regression on the covariates, on that SNP, and also on the mediator. So in the lung cancer example, the mediator will be smoking. In the asthma example, the mediator will be gene expression. And the second model is you regress the mediator, that is smoking, on the covariates and also on the SNP. Okay? So the coefficient of the SNP in this logistic model and this beta S, this measures the effect of the SNP on lung cancer through other biological pathway. And so this coefficient that measures the association between smoking and the lung cancer, this is the beta M, and the association between the SNP and the smoking is this delta in the second model. So the indirect effect is this beta S. And indirect effect is the product of these two in this simple setting. So why we found the SNP associated with lung cancer and also the SNP associated with smoking, why the mediation effect through the smoking is so small? The reason is even this delta, we know that based on epidemiology, the smoking effect on lung cancer is huge. This part is no, this beta M is big, huge. And so the GWAS study found out there is association between the smoking and lung cancer. And also GWAS study also found out there is association between the SNP and the smoking. So this beta is significant, but the problem is the effect of the SNP on the smoking is very small. Okay? So that's why when you multiply them together, and in order to find out this delta to be highly significant, the sample size is huge. Okay? So that's why you can see that by multiplying them together, the mediation effect is small. So most of the effects are through other biological pathways. Okay. All right. So also by using information of the STIP and the gene expression, one can increase the chance of detect association. So this is based on asthma data. So if one only uses GWAS data without gene expression, only 5 genes are significant across the genome with FDR less than 10%. But if one uses the gene expression data plus the GWAS data, and one can identify 50 genes and which FDR less than 10%. So by combining information of the GWAS data and the gene expression data, one increases the power. Furthermore, by using information of the GWAS data and the gene expression data, and then one can understand the disease causing mechanism. So this is analysis of the methylation data, microRNA data, and the gene expression data on the survival of glioblastoma multiforme. So you can see we can identify the association across all the different parts. And so for this particular gene, you can see that this is a p-value. So the path which has the smallest p-value is this one. So that helps we to identify which path and this methylation of this gene affect the 1-year survival. Okay, and so to conclude, so we are living in an exciting time, and for the quantitative genomics. And so moving ahead, we are going to make errors, and also we are going to do many trials, hopefully with a joint effort, and we are going to have a bright future. So in Chinese word, it's like basically across the river by touching the stone. And so before I conclude, I want to thank all the collaborators. And so the greens are my students and former postdocs, and they are in different institutes now. And also, before I conclude, because I know there are many students in this room, I want to do this advertisement for this book. And so the COP celebrated 50th anniversary in 2013. As part of the celebration, COPs, and so this called the Committee of President of Statistical Society. And so as part of the celebration, so we invited the past COPs awardees, and the COP offered 5 awards, and so probably the most well-known award is COP Presidential Awards. So that basically parallels the Fields Award in mathematics. And so COP also offer like a Fisher Award, a Fisher Lecture that's more for senior statisticians. And so we invited the past awardees and to contribute articles and as a celebration for the 50th anniversary. And so totally 50 people contributed. And this is a really great book for the students. And for those of you who are interested in knowing what is the cutting-edge research in statistics, and knowing the history, and knowing the personal stories of those well-known statisticians. And so this article covers wide breadths of the articles, including the reflections of statistical profession and statistical science, and also personal experience of the statisticians, and also the career advices for young statisticians. And so I think there are totally 6 people from Harvard contribute to this book, and 3 from Stat and 3 from Biostatistics. And also we negotiated with Chapman and Hall to make the book fully open access. So you can download this book for free and from the COPS website. So I want to just give you a few codes to stimulate the students. And here is just a few codes from the book. And so the first one is by Elizabeth Thompson. And so in her article on a journey with statistical genetics, she said, and think science, think positive, think why. And also in Larry Wasserman's article on the rise of machine, that is the rise of machine learning community. And so he said, if you cannot beat them, join them. Okay, so a few more quotes. And so this is from Don Rubin's article. I think this is a great article. And so in Don's article on the converting rejection into positive stimulus, and so he basically tells the young people about that all his rejections. And so he had many famous papers, and including like the EM paper, the causal inference paper, and missing data paper, except for that EM paper. And so he said 1% of his papers got accepted in the first time. And so for his causal inference papers, and so for those of you who know the literature, and he sent that paper to many statistical journals, all the top statistical journals, all reject him. And then finally, he got his paper published in a journal none of us heard about, in something like psychology journals. But that got a very high citation. And also for his missing data paper, it got rejected by a lot of journals and the statistical journal. Finally, he sent it to Biometrica. And so David Cox was editor. And then he invited Rod Little, who's his David former student, as a reviewer. And so David fell in love with that paper. And then later on, and Rod Little and Don, they became the best friend. And they wrote a book together. And so this basically tells the students that and tell the faculty it's okay to have a paper being rejected. And many well-known statisticians have paper being rejected. And also the other thing, and a good quote from his article is quality trumps quantity. And quality is more important. And also in Terry Speed articles, this is also a great article. And so I think this article comes from when Terry was too busy, he didn't have time to write. And then I sent him a few articles, and so tried to give him a sense about what's going on. And then after he read a few articles, and in particular, there's one article in Don's article. And so he mentioned when Don was young and was assistant professor, and one famous statistician, and he asked a famous statistician and how to be successful. And this famous statistician told him, be like me. And then Don said, so he modified that quote saying, with great people, like great mentors. And then Terry saw that article, and so he was afraid, like the junior people might be too scared. And then he said, well, do it your way, so be yourself. And so the final quote is from Bert Ephron. And so in his quote in this book, and his advice to the deans and the provosts is buy statistic future. Okay, thank you. Hope you have fun.
Do you have any opinion on whether the missing heritability is going to come from all the rare variants or maybe from other effects like nonlinear interactions between common variants?
Oh, yeah. And I think in theory, it could. And so in reality, we don't know. And because there's not, even for detecting the real variance effect, there's not enough power right now. So therefore, if one is interested in detecting the interaction, I think more and more data will be needed. I think it's very possible there are gene-gene interactions. Yeah, so right now, we don't have that knowledge. I know Jun has a paper on gene-gene interaction. Yeah.
Sorry, what was the reference? Who has a paper on?
What?
You just mentioned a paper.
Oh, Jun has a paper on gene-gene interaction. I think there are multiple papers on gene-gene interaction. Yeah.
Thanks.
Let me ask you, since you're chairman, would you accept the faculty to be promoted if the papers are all rejected?
Well, then I tell the faculty, so every paper has a home. I think this is what Rick Carroll said. Every paper at home, you just need to find it.
Okay, thank you very much.
Thank you.